groundbreaking research reveals the secret role immune cells play in building key brain cells in the womb. Did you know that immune cells in the human brain could be the maestros orchestrating the brain's development in the womb? New research suggests they trigger a dramatic increase in a crucial type of nerve cell. These nerve cells, known as inhibitory interneurons, make up a whopping 25% to 50% of the neurons in the adult cortex, the wrinkled tissue covering the brain's surface. In fact, our human cortex boasts more than double the number of these interneurons compared to a mouse's cortex. Interneurons are like the brain's traffic controllers, relaying signals between other brain cells and keeping that signaling in check with a chemical messenger called GABA. GABA is the brain's main inhibitory messenger, turning down brain activity by making neurons less likely to fire off signals. This balances out the excitatory signals that amplify brain activity. Issues with interneurons have been linked to various disorders like epilepsy, autism, and schizophrenia. But here's where it gets really interesting. Researchers have discovered a force driving interneurons to multiply in the developing human brain, and it might be unique to our species. That's why we cannot use traditional animal models, says study co-author Dongkun Yu, an assistant researcher at the University of California, San Francisco, UCSF. To uncover this mechanism, possibly unique to humans, the researchers grew a miniature 3D structure from stem cells that mimics a full-size structure in the human body, an organoid. Prior animal studies hinted at a link between an activated maternal immune system during pregnancy and fewer interneurons in their offspring's cortex. The culprit? Microglia, the brain's resident immune cells. In the past 5 years, scientists have started to recognize how the immune and nervous systems develop side by side. The microglia really fine-tune and regulate nervous development, says study co-author Dr. Xianhua Piao from UCSF. It really adds a new dimension as to how microglia exert their function. The team developed organoids resembling a temporary fetal brain structure that pumps out many cortical interneurons. They found microglia in their organoids were a key source of insulin-like growth factor 1, IGF-1, driving the rapid interneuron increase in early development. Turning off IGF-1 signaling blocked this increase. But deleting the IGF-1 gene in mouse microglia didn't change a thing, suggesting this chain of events could be a human-only phenomenon. These findings hint at an evolutionary adaptation of microglial function to support the increased demand for interneurons in the human cortex. In other words, it could be a feature of human evolution helping to explain our unique cognitive abilities. But remember, organoids aren't perfect replicas of the human brain. So far, the model is good enough for especially the proliferation stage, very early stage, says Yu. They don't capture later brain development stages or circuit-level activity. Future work could further clarify this newly discovered role of immune cells in the brain. And this is the part most people miss. Our understanding of the brain is constantly evolving, with each new discovery opening up a world of possibilities for future research and potential treatments. What do you think about this groundbreaking research? Do you agree with the findings, or do you have a different interpretation? Let us know in the comments below.